this computer. Okay, here we go. Let's talk about malaria. This is a fascinating topic, and it's very detailed. A lot of information here, and the life cycle is very complicated, but it's a very interesting pathogen and disease. So let's begin. Malaria. So let's first talk about the evolutionary history of Plasmodium. Plasmodium is the name of the pathogen that causes the disease malaria. And there's an interesting thing about Plasmodium. Plasmodium has two ways of reproducing. So Plasmodium is both sexual, and it's sexual in the mosquito, and it's also, it can reproduce asexually in the human. So it's a very interesting organism. It's a single-celled eukaryote. We would classify it as a protist. It's very interesting in that it has both a sexual cycle and an asexual cycle, and it combines these. So we know that malaria is or Plasmodium. When I say, I'm probably going to improperly say malaria and Plasmodium interchangeably. Of course, you should just know right now that Plasmodium is the pathogen that causes the disease, which we call malaria in humans. So I'm probably going to say these interchangeably, which is technically incorrect, but just FYI. We know that Plasmodium is very, very old, and there are fossilized amber fossils of dipterans, so flies. In this case, I think it's a biting midge, and just like Jurassic Park, they can take these fossilized amber samples and they can do microscopy. And there's papers where they point out these blobs. I am not like a paleontologist, so I cannot like assess like the rigor of this data, but they claim that these are malaria parasites, Plasmodium parasites in this biting midge, in these old fossils. So we know that the association with dipterans is extremely old because we can see these in these fossils through these microscopic techniques. And you might ask yourself, it's interesting that in that malaria, Plasmodium, it's sexual in the mosquito, but asexual in the human. So you might ask the question, where was it first? What was it always? Oh, here's Daniel joining us. Was it always in? I just have to minimize you, otherwise I'll become distracted. You can ask a question: was it always in one or the other, or was it always in both? Like, this is such a strange life cycle that it kind of facilitates the question, where was it first? Was it first a sexual organism in the mosquito, or was it first an asexual organism in a human that then learned to become transmitted by the mosquito? And the answer is probably that it was first a sexual organism that was surviving in dipterans. I talked to Dr. Hardy about this question when we were first preparing these lectures. And so the rationale for this is that he said, sex is very hard to evolve. It probably evolved only once. Conversely, it's very common to lose sex. So the idea that like there is an asexual stage of this creature, it was probably ancestrally sexual in the dipterans, and then it learned to become asexual when it was in the human. So that was probably the order of the evolutionary events, although I can't say that for sure. That's my hypothesis. And there's different sort of terminologies. There's lots of terminology with malaria. It kind of gets ridiculous, but I'll point to the ones that I think are important, and I'll point to the concepts that you kind of want to know. So you'll see this term gametogeny. That means it's in a stage where it's making gametes. Okay, that means it's in, if you say gametogeny, that means it's in the sexual phase in the mosquito. If you see schizogeny, schizogeny. That's the asexual stage where it's reproducing in the human. Yes. So there's many different plasmodiums. So here's a phylogenetic tree that's kind of showing you the evolutionary history of different plasmodiums. But there's really only about 6 plasmodiums that we care about as humans, because these are the ones that really cause pathogenesis in humans. So there's Plasmodium vivax, Plasmodium knowlesi, Plasmodium malariae, and Plasmodium ovale. And there's 2 different variants of ovale. Okay. And these are 1, 2, 3, 4, 5. These are 5. Okay. And these 5 are considered the older malariaes, the older plasmodiums. And these ones are less pathogenic to humans. They are pathogenic. You will get disease from these 5 older plasmodiums, but they're usually not the malarias that are killing people. The one that's killing people is usually Plasmodium falciparum or falciparum, depending on how you say it. This is the 6th one that we care about. This is usually the one that's killing people, and this is the one that's thought to be kind of like newer, quote unquote newer, although again, like that's probably like a bad evolutionary term. They're all extant, so they're all sort of like equally new in theory, but these are the ones that are sort of like newer in the relationship with humans, and that's probably why they exhibit a stronger pathogenesis, because humans have probably like been exposed to these ones for a longer time, and so they've in some ways become kind of attenuated. The reason that falciparum is really bad is that it can invade many different types of blood cells and many different cell types. So what's happening in malaria is you have this pathogen, call this the plasmodium, and the plasmodium will infect your cells. Many times it infects what are called I'm going to spell it wrong, but erythrocytes. Okay, these are your red blood cells. And Plasmodium falciparum is unique. It can infect many different types of cells. And what's happening is it's coming in and it's replicating, and it's replicating itself so much in the schizogenic cycle that it replicates almost like a virus, and it replicates so much that it just explodes your cells. Okay. And so if you can imagine, if falciparum is able to invade many, many more different types of cells, it's exploding more different types of cells in your body, and hence that's why it's more pathogenic. Whereas the older ones have kind of like calmed themselves down a little bit. They're not replicating as much. They're not invading many of the different types of cells. And we talked about this in some of the earlier lectures, that this was actually like a pattern, that the pathogen does not want to kill the host. Why? Why does the pathogen not want to kill the host? Yeah, they want to get the nutrients from the host, but think about this more. Why do pathogens not really want to kill the host? It's not really in their interest to kill the host. Why?
Because their life cycle depends on it.
Yeah, so I heard 2 answers. Brian got it right. I heard Daniel say something, but I couldn't catch it. Brian suggested that they don't want to kill the host because the host is like their vehicle, and they want to be able to spread to more humans. So if you just instantly kill a human, that human is not around anymore to be spreading that malaria. Whereas if you can keep the person alive and sort of like learn to live inside that person without killing it, you actually are able to spread your genes more effectively. So pathogens in general do not want to kill the host. This is sort of like a byproduct. I think I'm sort of guessing a little bit here, but this is sort of like a byproduct of in falciparum, that it hasn't really learned how to evolve. It hasn't evolved yet how to be effective in the human body. It hasn't learned how to like keep control of itself, if that kind of makes sense. Okay. So just a couple details about some of these other ones. Knowlesi, Plasmodium knowlesi is a zoonosis. Its reservoir is monkeys. So it floats around. It's a malaria that floats around in monkey blood, and it can every once in a while get transferred to a human. The other malarias are not typically considered zoonotic because most of them are human to human transmissions. But there are cases where malaria is clearly zoonotic, and also this is an area of active research. So it's an area of active research. Many times what people are doing, and I know Dr. Sarzotti actually does work on this, where they'll go to countries where malaria is endemic and then survey blood from different animals in the population to be able to see where those malarias are sort of hanging out in reservoirs in those populations. And they are able to find, I think if I remember correctly, I think there's examples where you find vivax in cows. So I essentially want to say that how much malaria is zoonotic, like quantitatively how much, that's an area of active research in comparison to how much of it is transmitted human to human. That's an area of active research, and it probably is different depending on where you are. So there isn't a set rule. Vivax is really unique. Okay, so there's kind of ones you really want to remember. You really want to remember Vivax, and you really want to remember Falciparum. Falciparum, what you remember about it is that's the deadly one, that's the newer one. Vivax has some really unique attributes about it, in that Vivax can hide dormant in your liver in this special stage called the hypnozoite stage. So when we go into the malaria life cycle, coronavirus, when we go into the malaria life cycle, you will see that there are stages that infect the liver, and Vivax has learned how to go into the liver and sort of hide there and stay dormant. And again, you can see why this is advantageous, because the Plasmodium does not want to kill the human. It just wants to sort of like be a parasite that survives inside of it. So if it can hide dormant, like a virus can hide dormant, that's advantageous to the Vivax. So the Vivax has evolved this unique ability. And actually, you'll see this in the epidemiology of Vivax, in that Vivax is kind of like a world traveler. It travels all around the world. And Falciparum is sort of not as good at traveling around the world. So the other thing about Vivax is what will happen is you'll go through stages where you will exhibit the disease, and then it will go away. And then I said this earlier at the beginning of class, when your immune system is attacked by something else, maybe like a virus or maybe like a separate pathogen, then your immune system becomes weak and the Vivax can come back up, and it sort of comes back out at that time. Okay. So we talked about this in the mosquito lecture, but just to re-emphasize, only Anopheles mosquitoes spread malaria. Okay, and Anopheles mosquitoes were the older sort of architectural structure of mosquitoes. Anopheles mosquitoes were thought to be kind of like the more older mosquitoes, as opposed to Aedes and Culex. Aedes and Culex do not spread malaria. So you should know that. Anopheles are the ones that are important, and the most important one is Anopheles gambiae. That's the one that likes humans. That's the mosquito. It's evolved for human blood, and it spreads Plasmodium falciparum. So the history of malaria, again, one reason I like this class is actually like a lot of history related to these diseases, and malaria is no exception. Malaria was first documented by humans about 2,700 years ago in Chinese documents that sort of describe a disease that seems like it's like malaria. So it's been around for a long time, and we've been sort of documenting it for a long time. The word malaria means bad air. People always knew that it was associated with like swamps and wetlands, but nobody had any idea that the mosquitoes were spreading it. They originally thought that the swamps like give rise to like bad fumes and toxic air. And if you breathe this bad air, you get this disease called malaria. So this was before we knew about germ theory and before we knew that mosquitoes spread the disease. That's how it got its name, bad air. And in 1623, there's an interesting incident where malaria, so if you know anything about Catholicism, there's the leader of the Catholic Church, which is the Pope, and the Pope sort of has control of all the Catholic dioceses. And in 1623, the Pope died from malaria. And when the Pope dies, there's a conclave where the cardinals come together and they elect a new Pope. And during the conclave, after the Pope died of malaria, 6 cardinals also died of malaria. So there was an outbreak of malaria that changed sort of like the structure of the Catholic Church. And of course, around this time, the Catholic Church had significant influence, political influence on world events. So these were considered significant events influenced by malaria. So the things you want to know about malaria is it's an epidemic disease. So epidemic diseases, that's a special type of disease in the sense that, so I should just write this out. Epidemic diseases are special in that they're famous for being able to explode. Like they all of a sudden come from nowhere and then they just explode. And then all of a sudden, many people have it. So malaria is an epidemic disease. It can explode. And what you'll learn about malaria is as you study these explosions of malaria throughout history, they always associate with patterns in almost like human biology. It's sort of like human biology that facilitates human behavior that facilitates the explosion of these explosions of malaria. And so what you'll often find is that famous malaria explosions occur coincidentally with mass migrations of people. So in instances where there's many people all of a sudden moving from one region to another, what happens is usually some of those people have the parasite inside their blood. They move to a new place where all of a sudden now there's a vector that can transmit that pathogen. And now since they've moved, those people who have that disease are now in a context where they're also mixed in with people who have never been exposed to that disease before. And now all of a sudden the mosquitoes start picking it up and it explodes throughout a different population. So that's sort of a very common pattern that you'll see when you study the explosions of malaria throughout history. Malaria was originally not in Europe, so malaria comes out of Africa. And the way that it got into Europe, we actually know, we actually kind of know the route in which malaria has spread to Europe. So in Italy, so here's Italy, this is Italy. And if you know anything about Italy, Italy was famous for the Roman Empire, right? And Italy is geographically, whatever, it's like a boot or something like that. Italy is like a base in the Mediterranean, right? And if you cross the Mediterranean, down here is Africa, and up here is Europe. So Italy is kind of like this hub of transport through the Mediterranean. And in the ancient world, Italy and Rome specifically was a very big place that was sort of having lots of movement through it, okay? And aside from Italy, there's a little island called Sardinia. And Sardinia is thought to be the sort of like the foothold through which malaria came from Africa, established itself in Sardinia, and then from Sardinia spread throughout Europe. Okay. And this is actually associates with famous ancient events. So originally the Carthaginian, can't even say it, from Carthage, the Carthaginians were Africans. They originally conquered Sardinia. Okay, so when they conquered it, they brought malaria to it inside of them. And then what they started doing is they started essentially doing agriculture on Sardinia. And another pattern that you're going to see with malaria is as people start to do agriculture, cultures obviously start to recruit other people into forced labor, forcing them to do the agriculture for them. So what happened is after the Carthaginians conquered Sardinia, they started using it as a hub for agriculture, and then they started importing slaves into Sardinia, which is then importing more and more malaria into Sardinia. Okay, and then you can imagine what's happening here. What's happening here now is a chain of events, and you see this repeat throughout history. Is one, what happens is agriculture starts, okay. Two, what happens is in order to make agricultural land, you need to clear the land. Okay, so they clear the land. That means they cut out the trees, they get rid of the bushes. All of a sudden, what happens when you clear the land? You start to create swamps. When you clear the land and it rains, you now have swamps, okay? And then now what you have is you're also importing people for forced labor. So now what you have is you have a drastic increase of reservoirs, okay? And you have a drastic increase of swamps, which facilitate a drastic increase in mosquitoes, right? So now all of a sudden you can see how this is sort of like a nasty cycle. And so what happens is there's more and more reservoirs, there's more and more mosquitoes, and the malaria just sort of like cranks up and becomes endemic in these regions. So this is a negative, we call this in biology a negative, spell this right, a negative feedback loop. This means like conditions keep getting worse and worse and worse and amplifying each other. And so Sardinia was then later, so Sardinia at that point became sort of like endemic for malaria. And then it was later conquered by the Romans. So the Romans then conquered Sardinia, and they did the same thing. They were importing slaves and ramping it up. But now there's more and more movement up into Europe, right? So now that's the foothold through which malaria sort of established itself in Europe. Okay. So that's the history of the expansion of malaria out of Africa. And it's actually, again, more interesting tidbits about the history is when the Romans had Sardinia, the Romans knew how horrible it was there because they had created this hell from this negative feedback loop. So when the Romans had it, it was actually considered like a penal colony, which is kind of like where when you commit a crime, and they don't want to sentence you to death, but they do want to sentence you to death, then they send you to Sardinia and they know you're just going to work there until you get malaria and die. So it was actually used as like a death sentence, which the Romans used to sentence their criminals. They would send them to Sardinia to die a horrible death of malaria. Okay, and then because humans are not very good creatures, patterns repeat themselves over and over and over in history. Okay, so then what happened is malaria spreads throughout Europe. And again, you see different malarias spreading. So vivax, because it can kind of like hibernate, is really good at spreading into different climates. So vivax was really able to spread throughout Europe. Falciparum is not as good as at spreading out of tropical climates, okay? So falciparum kind of isn't as spreading as much, but vivax really is spreading sort of like everywhere. And then of course what happens in the 1600s, 1700s is the New World is discovered, okay? And of course, agriculture starts in the New World and they start importing slaves into the New World. And when slaves were imported into the New World, there were two sources of these slaves. So when they first got into the New World, again, geography bad here, but here's Florida, here's Texas, and then there's all these islands, there's Cuba, Jamaica, and then there's all these islands. This is the Caribbean, okay? When people first got here in the 1600s into the Caribbean, they forced these people who lived here in the Caribbean into becoming slaves. And then the slave trade started in North America by North Americans buying Caribbean, what you would call Native Americans of the Caribbean, okay? And so at this point, there was no malaria being imported because the people here did not have malaria, okay? So when the United States in its early days was buying slaves from the Caribbean, there was still no malaria in the United States. But then there was some kind of law where the law was like, they didn't want any more, they either ran out or they didn't want to. I don't know the history of the slave trade very well, but there was some point a decision was made where they're not going to buy slaves from the Caribbean anymore. They were going to import them from Africa. And then at that point, you have the importation of slaves from Africa, which of course bring with them vivax. Okay, so vivax in the Americas, the reason it established itself in the Americas was because of the slave trade. And then falciparum comes later. And the reason falciparum comes later is it's not as good at moving, whereas vivax can come right away because it's sort of like hibernating inside of people. And this is interesting. We can actually, like, the reason these are hypotheses. We don't know this for sure, but one of the reasons we think that this is true is because there's actually documented evidence of when slave ships got to America, and then there's actually like what would you call it, like ship manifests that have this documented. And what happened was there was a large shipment of about 30,000 African slaves and into South Carolina. And right when that happened, that coincided with a gigantic outbreak of the worst malaria they had ever seen in South Carolina. And so that's why they suggest that was at that point they think falciparum made it to the New World. Okay. More about, because we don't quite hear this history, because the United States now does not have malaria anymore. So it's kind of like we had it in the past, and so you don't learn a lot about this history. But the reason also, there's many, many more reasons why the United States was sort of like a perfect breeding ground for malaria. At this time, when we were importing the slaves, much of what we were cultivating was rice, okay? And if you know anything about rice cultivation, essentially what you do is you build swamps where the rice grows. So wherever there's rice growing, there's obviously mosquitoes. So that makes matters worse, okay? And then the anopheles mosquitoes grow in the rice paddies. That's the thing that we already had here. In the United States, we already had anopheles vectors. So we already have anopheles mosquitoes. They did not need to be imported. All we needed was just the delivery of the pathogen. And then that facilitated outbreaks. And again, you see these outbreaks of malaria documented where you see large amounts of mortality in people under the age of 20. That's characteristic of falciparum. That's another reason why we think we can see sort of like when falciparum arrived versus when vivax arrived. And one of the interesting things is, again, so this is like sort of like getting what they deserved. Well, we don't know this for sure because the African deaths would have not been reliably reported, but it's many of the Caucasians that are dying because in many cases, people from Africa have evolved such that they can live with malaria. Whereas if you're Caucasian and all of a sudden it appears in your local community and you've never been exposed to it before, you're the people that are dying. So it's kind of like they're the ones that are doing this, and they're also paying a cost for it. Although they would have had no idea that this was mechanistically happening. It's probably the demons or the witches.
I have a question.
Anyway, okay. Yes, we got all that.
I have a question.
Your question, yes.
So this is a very common statement. America claims that they don't have malaria, right? My question is that how is it that they can prove they don't have malaria? Like malaria has been eradicated in America because there's a lot of international visitors. So at some point you would expect malaria to
Okay, so let me say your question so that everybody here can hear it. The question was, everybody says in America that malaria is not in America. How do you prove that malaria is not in America? So if you're asking about actual parasites, There's nobody going around and like PCRing or checking for like malaria in America. I mean, maybe, I'm sure, actually I should not say that, because I'm sure there are. Like, I'm sure there's like CDC people who are like doing PCRs to make sure we don't have malaria. But essentially, like nowadays we know that because if there are certain cases of diseases that doctors find, if it's an epidemic disease, they're forced to report that to the CDC. So the CDC in America collects numbers for how many instances of particular cases, okay? And when there's a case like malaria in America, the case is investigated, and so they determine what did that person just come here from a foreign country? Is it possible that they got malaria from the foreign country, or is it possible that this is like a residual situation where vivax is coming back up and the infection was already there? Does that make sense? So we keep track of these numbers through the CDC. Does that answer your question?
Yes, it does.
Okay. Okay, so just patterns, again, just patterns of malaria, wherever there's movement of people. So it's the biology of humans, it's the behavior of humans that facilitates malaria outbreaks. And wherever there's large movements, just what tends to happen is there's movements of new pathogens into new areas. And if there's vectors that can transmit those pathogens in the new area, then the transmission will occur. So when malaria outbreaks occur, it's usually the case that there was a large migration. So now this relates to Daniel's question. A very common question that I get asked all the time is, so why don't we have malaria in America now? What was done to, since we have all the conditions in which malaria can survive here and thrive here, we know that it was thriving during the Civil War. What happened that caused malaria to decline in the United States? So many different things all at the same time. One, we started growing rice in a different way. So we started, instead of irrigating with essentially swamps, people started using tidal irrigation. So it's where you're by the ocean and the tide brings in water and then it recedes. And that creates conditions where the mosquitoes can't thrive in those environments because the water's always receding and coming back. So we started switching the way that we were cultivating rice. Two, windows were invented. So people started to sleep indoors with windows. And if you have windows, you don't have mosquitoes coming in to bite you at night. Okay, that's an obvious thing. Three, a new drug became available. So quinine. Quinine was an effective treatment of malaria, and that sort of helped it. And that's, all these things are coinciding at the same time. Four, agriculture begins to shift away from forced labor after slavery was outlawed, and then also too just the technical revolution, like technological revolution, making agricultural machines. People are using tractors now and things like that, as opposed to humans doing the actual labor of agriculture. So the fact that machines are now being used means that there's less reservoirs for the malaria to hang out in, and that facilitates it going away. So mechanization. And then there was an interesting event in 1890 where there was a boll weevil outbreak, and that reduced the cotton acreage in the South by 50%. So essentially, like many farmers of cotton went out of business in the 1890s because of this boll weevil outbreak. And after that, like it didn't recover as much, and it was never the point at which there were humans doing that labor after that. We began to establish public health departments. So each town kind of has a vector control department that's associated and reports like within the Department of Public Health. And a particular goal of public health movements is to eradicate vectors. So we talked about that idea by Patrick Manson, that you could kill the mosquitoes to prevent transmission of diseases. That sort of was really put into effect. And then 7-DDT, which is an insecticide, was used very heavily in the late 30s and the early 40s, and during and after World War II, DDT was used extremely heavily. And whether you like DDT or not, it's extremely effective as an insecticide, as long as you don't have resistance, which we do have a little bit now. So all these things contributed to the decline of malaria, and with this is why we can say now, like, we don't really have malaria in the United States. although we have the conditions under which it could come back. I went through this. Windows, bed nets are a big thing. We also have sort of like, after the Civil War, the government of the United States kind of became quite stable. And so for the last few hundred years, we've had sort of stable government. And as I think, another pattern you'll see is governments that are stable usually have effective vector control programs. Okay, so malaria today. Again, like filariasis, we have reason to be optimistic because malaria consistently every year gets better and better and better, in a sense that there's less cases each year and there's less people dying from malaria each year. Okay, so when I was a graduate student, I would read papers that would say every year about 1 million deaths every year from malaria. Now you read papers and it's less than 500,000 usually. And I know this because like these are always what are the first sentence in people's grants is how many people are dying of malaria every year. So we're getting better and better and better at sort of reducing transmission of these diseases. And I would say that this is a real cause for being optimistic, that people always like to think we live in the worst times and the world is horrible, but in many respects, the world is actually getting better and better and better every year. And yet it's still a problem that we need to work towards dealing with. So we went over this person last time, Patrick Manson, father of vector biology. He was talking to Ronald Ross. I won't talk about his filarial discoveries because we talked about that last time, but he was talking to Ronald Ross, this gentleman who was a researcher on mosquitoes. And Ronald Ross was the first person to demonstrate that anopheles mosquitoes can transmit malaria, and he did it in birds. Okay, and the reason he probably didn't do it in humans is because it's unethical to infect. It's unethical to do that experiment in humans. But nevertheless, it was later discovered by this person, Alphonse Leverin, that the malaria parasite is in the blood and it can be transmitted. This person, Giovanni Grassi, was the first person to prove that mosquitoes can transmit malaria human to human in 1898. So there's kind of these cycles of discovery. And again, like you want to remember Ronald Ross. He actually he won a Nobel Prize for this discovery that malaria is transmitted by anopheles mosquitoes. And he was in contact with Patrick Manson throughout this time. What was this? I'm just looking quick. What was this person's discovery? This person, Alphonse Leverin, was a person to see the parasite in human blood. And we'll talk about what he was seeing, which is called exflagellation, which is when the parasite becomes so abundant in a blood cell that it explodes the cells and pops out into the bloodstream. Okay. The malaria life cycle is really complicated. And it's complicated because malaria is sometimes sexual, sometimes asexual, and it has a cycle in a mosquito. It has a cycle in the liver, and it has a cycle in the blood. So its life cycle is extremely complicated. This is something like, my analogy for this is kind of like glycolysis. Like when you have to, in biochemistry, you have to memorize like all the enzymes and then you just kind of like forget it. There's basic things that you want to remember about this, which I'm going to try to emphasize here. Don't get too stuck up on kind of like terminology and the names. Every time I need to remember the names, I kind of just like Google it and just like quick make sure. But you want to remember the general concept of what happens throughout the cycle of this disease. So we'll start here. What happens is a mosquito takes a blood meal from somebody who's already infected with Plasmodium, okay? And what they're picking up are the gametes, the gametocytes. That means these are haploid and they're ready to mate. Okay? And so what happens is this is now, at this point, this is in the mosquito gut. In the mosquito gut, mating occurs between the gametes. So there's sex, this is where the sex happens in the mosquito, okay? And then what forms is this creature called the ookinete, which is diploid. Make sure I get that right. Ookinete, diploid, yes. So the ookinete is what's produced after the sexual replication. And then what's happening here is in the mosquito gut, so let's say this is the mosquito gut. All guts have a layer of cells, which is like the wall protecting inside from outside of the mosquito. I think I've drawn this. This is also true in humans, it's true in all organisms. There's a layer of cells called the epithelial layer of cells. And if inside is the gut lumen, in here is the body cavity. This sort of wall right here is called the basal lamina. And this is sort of like the last line of defense, preventing stuff from here just sort of freely diffusing into the body cavity. So it's like a wall. And what happens is the ookinete, it invades this cellular layer of epithelial cells and it starts to bulge, okay? So if this is like the ookinete, it invades and establishes itself right here and it starts to create this like bulge in the basal lamina. And that is this, the oocyst. And what's happening is it's now replicating itself asexually, okay? It's replicating and it's creating sporozoites, which are the asexual daughters and sons. I don't know about the sex and daughters and sons when I say that, but they're the asexual offspring of the ookinete. And what happens is it just keeps replicating, and then all of a sudden it explodes beyond the basal lamina into the hemolymph of the mosquito. Once it gets into the hemolymph of the mosquito, these are called sporozoites. The sporozoites move to the salivary glands of the mosquito. Once they get into the salivary glands, they get spit out when the mosquito bites a human. This is the transmission from human to mosquito. Okay, now this is only half the life cycle of malaria. So the asexual life cycle of malaria. Once it gets injected into the human, we start here. Okay, so we'll start here, it gets injected into the human. Now the malaria moves through the blood straight to the liver, and liver cells are called hepatocytes. You'll see this term a lot. You do want to remember that. Liver cells are called hepatocytes. It infects liver cells and it replicates inside the liver cells and it explodes the liver cells. And it does this for like a period of time, okay? So what you're starting to notice is in the human, there are stages where the malaria is inside of cells and there are stages where malaria is outside of cells, okay, in the blood. And this becomes important for the pathology. So there's a liver stage. At some point, these schizonts, these are called schizonts, at some point they decide, okay, now they're gonna colonize and more heavily colonize the blood because their goal is to become gametocytes and eventually get back into a mosquito, to get transmitted to a new island. So that's why they wanna move into the blood. So at some point they move out of the liver into the blood, into this new cycle, which is called the erythrocytic cycle, which is where they're now, instead of invading liver cells, they're invading red blood cells, doing the same thing, replicating, and then exploding the red blood cells over and over and over and over again. And they produce gametocytes, which now you remember are the creatures that are picked up from the mosquito when the mosquito bites. And at this point, here's why I was saying that about the sex. At this point, gametocytes, now they have their sex either male or female. And once they get picked up in the mosquito, they mate and the cycle continues. So a couple things, let's talk about this. One of the things that you see with malaria very commonly is you see these recurring fevers, okay? So what do you think the rationale for those recurring fevers are? So you see like periodic fevers where people will have like a fever for like 3 days and then they'll like be okay. And then later they'll have another fever for like 3 days and then they'll be okay. And this will continue.
It's because of the
what?
It's because of the viral load. The viral load.
Well, it's not a virus. It's not because of the viral load. But what's happening is what you're actually seeing with the fever is what you're seeing is this stage right here. When the schizonts rupture the red blood cells and pop out into the bloodstream, that's where you see the fever. And then what happens is at some point they stop doing that and they go back into red blood cells and just kind of chill out, or they go back into the liver and chill out. And so you see these cycles of fever, and the cycles of the fever are when they're rupturing the red blood cells. And that's called, the name for that, it's called exflagellation. That's called exflagellation. And that's what the one person saw when he saw malaria in the bloodstream. Okay. So again, you kind of just want to know the gist. You want to know there's a cycle in the liver, there's a cycle in the red blood cells. The reason it's in the red blood cells is because it wants to get picked up by a mosquito. Once they're picked up by a mosquito, they mate, they move outside of the gut, get into the salivary glands, and then get transmitted to a new human, and the cycle continues. Okay. This may or may not be worth going over. Again, like there's so much about malaria, like we could go into the nitty gritty details of like how it's invading particular cells. I have a slide here because I show you that there's in each cell has like a particular kind of like its own skin. And there are special glycoproteins, which are proteins that have sugars attached to them, which the malaria parasites recognize. So if you're wondering like, how does the malaria parasite get inside of like a liver cell? Or how does the malaria parasite get inside of a red blood cell? It's blind, how does it know how to get in? It's got receptors. These receptors bind to certain glycoproteins on the skin of these particular cells. Once that reaction happens, it gets endocytosed and then it's inside the cell. Okay, that's how this sort of process happens. I talked about all this. Talked about it. This is the full picture, so you can see how complicated it is. I don't remember all the terms, but I do remember usually the basic gist. I think I just put this in because it's a pretty picture. Okay. Let's talk about sort of like the epidemiology of the disease and which factors contribute towards this epidemiology. I'm not able to draw. So if you have more contact between humans, it's going to be more likely, and you're going to be in an area where you get malaria. So malaria is going to be easier to spread in cities with high populations, right? Because the mosquitoes are more likely to find somebody who already has malaria and then more likely to get a second blood meal. If you live by yourself out in the jungle, it's unlikely you're going to get malaria unless you're getting it from a zoonotic source, like a monkey or something. Okay, so if humans are spread apart, you don't usually end up with malaria. Age of the mosquito is extremely important because there's a time, the extrinsic incubation period, there's a time for which it takes the malaria to go through that mating process, go through the ookinete, get into the salivary glands. There's a time, okay? So that if the mosquito is very old and is taking a second blood meal, then you're at risk of getting malaria. You're not at risk of getting malaria from a mosquito that's never bit before or a young mosquito. And malaria is unstable. So we could, in theory, make malaria extinct because there are many, many documented cases. Like I said, it's epidemic. There's cases where it explodes and then it goes away. And the reason it goes away is because if the population of people who are carrying the parasites goes below a certain threshold, then the probability that a mosquito that picks up malaria will feed a second time is below the actual probability that will actually like occur. So malaria is actually one of the few diseases we're working with that we could actually quite easily eradicate. We could make it extinct because it periodically goes extinct in regions after these epidemic explosions. We could make this happen. I talk about this. This is a special thing which is especially exemplified in the malaria life cycle. There's this, in vector biology, there's this hypothesis called the bottleneck hypothesis, which is if you look at all these vector-borne diseases, they have all these complicated life cycles. Every single one of these points in the life cycle is like a link in a chain. And if the parasite doesn't complete this link, at some point, if you cut that chain, you can stop transmission of the disease. And now you can understand that there's going to be certain chains that are going to be easier to cut than others. Okay, so the bottleneck hypothesis is kind of like in vector biology, we understand that these life cycles are like chains. And if we identify the weak points in the chain and we target those weak points, we might have a better chance of eradicating the disease. So with malaria, the weak point in this chain is this ookinase. Okay. So if you look at, this is a tough figure to look at, but if you look at here, the parasite number, this is a parasite number in mosquito. Gametes, so this is how many gametes a mosquito picks up from an infected blood meal, about 450. How many of those 450 get to form a zygote, meaning they mate? About 13. How many go from zygote to ookinase? About 6. How many are actually able to form the oocyst? About 2. So essentially the weakest point in the chain is the stage of the oocyst. If you can just block 2 oocysts from exploding, you've prevented malaria from spreading. Does that make sense? So essentially what this is suggesting is it would be much easier for us to disrupt malaria transmission in the mosquito than in the human. Because in the human, the numbers of parasites is going to be vastly higher than 450, 13, 6, and 2. It's going to be vastly higher. And it's even more complicated in the human because there's stages that are intracellular and stages that are extracellular, and your antibodies cannot find parasites that are intracellular. So it's kind of like effectively hides. So it's a lot more difficult to deal with malaria in the human, in theory, according to this bottleneck hypothesis. We'll finish up here soon. The symptoms of malaria: headache, fever, fatigue, pain, chills, sweating. The fever recurs. And you also get, the reason that malaria can kill you is because you get anemia. So what happens is when it goes into your red blood cells, it will destroy your red blood cells. And if you can't produce enough red blood cells, you will then starve yourself of oxygen. Okay, so this is how it kills you, is it kills your red blood cells. Your red blood cells carry oxygen to the organs in your body. If it kills enough of them, your body literally does not have oxygen and it dies. Yeah, you get organ damage. Quickly, we'll just, and I just wanted a couple quick things. So there's a very, very famous, one of the reasons we know that Sardinia was so rampant in malaria is because we can see the effect of selective pressure of malaria in the genotypes of people who come from Sardinia. So sickle cell, this is a famous mutation, sickle cell anemia. Sickle cell disease is a mutation in hemoglobin. Hemoglobin is a protein, a little machine in your red blood cells which binds to oxygen. So hemoglobin has iron groups. Iron binds to the oxygen. Hemoglobin is actually the protein that's carrying oxygen and delivering it to the parts of your body. And if you have the sickle cell mutation, it's a single SNP, an A to a T, that causes a change in amino acid from glutamic acid to valine at this particular position in the beta chain of beta hemoglobin. And if you have 2 copies of these allele, it's bad. You get the disease, you get this. You get mutated red blood cells. But if you're heterozygous, so you just have 1 copy of the mutant hemoglobin and 1 normal copy, your blood cells look like this, and you're resistant to malaria. So being heterozygous for this mutation is actually quite good. And you can see how if you have endemic malaria on a place like Sardinia, it would start to select for this mutation in heterozygous individuals. Okay, and you can actually see this. So we can actually see this in the genomes of people who come from malaria endemic places. We have effective drugs that treat malaria. We have quinine, which comes from the South American cinchona tree. We have other treatments. Chloroquine, which you've heard about a lot in the coronavirus stuff. Chloroquine was originally, the reason why people were so excited about chloroquine for coronavirus was because this was a drug we have a lot of experience with, because people have been using it for many, many years to treat malaria. And the reason it works in malaria is it's thought that as the malaria parasites invade red blood cells, the hemoglobin can actually be toxic to the parasite. And it's thought that chloroquine inhibit. So what happens is the parasites, they're able to crystallize the hemoglobin and kind of put it into inclusion bodies. Chloroquine is thought to inhibit that crystallization process. And that's thought why it kills malaria parasites. We also have this drug called artemisinin. This is she won the Nobel Prize to UU. She won 2015 Nobel Prize. And what she was doing was she was reading ancient Chinese literature on ancient Chinese herbal treatments for diseases. And she found out that one of these plants actually had an active chemical called artemisinin, or at least this chemical was derived from that chemical, which is extremely effective for treating malaria. So she won a Nobel Prize for this. Last slide. This is an interesting slide. In the old days, when you got syphilis, it was thought that syphilis was much worse than malaria. And so they could cure syphilis. One of the only treatments to effectively cure syphilis before antibiotics was they would infect you with Plasmodium vivax, and you would get high fevers, and the high fevers would heat shock your body to the point where it would kill the syphilis spirochete. So you could actually cure people of syphilis by giving them malaria, and it was thought I'd rather have malaria than syphilis. So they would do this. That this person is not a good person to follow, as he was a Nazi eugenicist. But just interesting, he was a person who won a Nobel Prize, so random weird things in science pop up and are interesting. So I end with that. Any questions? Okay.
Louis, take notes.
What?
I said Louis should take notes.
Okay. All right, goodbye.